Today we have Ross Smith. Ross Smith has a PhD in geology from Cambridge University, specialized in the sedimentology of the deep water depositional system. In a long industry career, he held a principal technical expert and chief geoscientist in role in Shell. He returned to academic as adjunct professor of the geology of the University of Malaya in Kuala Lumpur. Recently years, he has been making extensive use of the microtomography to solve problems in paleontology and mineralogy. So welcome, Rose Smith, and I will give you the stage to start your presentation. Once you would like me to move the screen, please let me know.
I will shout. Shukran. Thank you very much. Yeah, sorry for this late delay, but the talk's going to be about this technique of looking at things in 3D, microtomography, and specifically on amber, which is a kind of miraculously wonderful substance for preserving incredible details of ancient life. So thank you very much for the invitation. I've put the GSO symbol down there, University of Malaya on the right, where this work was mostly done. Let's go to the next slide. Okay, so this was a bit of a background that was already summarized there. So I started off as an academic in Liverpool University. I thought I'd have a couple of years in industry before going back to academia. And then it took a whole third of a century before I finally did go back to academia in University of Malaya. And you will see there's that one spell, move the cursor to the left, we see Oman. This was my time in Oman, which was a joy. And I was telling various people what incredibly fond memories I have of that country. What a special country it is. Now let's go on to the next slide. Please. Next, yeah, perfect. And here I'm just showing something, so that this is to do with a key theme of my entire career, which was making 3D models. And the idea was to co-visualize data, interpretations, and models, and also to have the ability to zoom out for context and zoom in for detail. And I've often found that problems at one scale are strongly helped by observations at a different scale. So that's the Ormond Langer Reservoir. These are deep water rocks. And then the bigger image at the back is the entire Norwegian Sea, so 500 kilometers by 250 kilometers by 4 kilometers. And there's going to be, in one of the stories I'll tell you tonight, nothing to do with the subsurface, but there'll be this same idea that you can make observations at one scale and learn something about a different scale. So let's go on to the next slide. And there's an obvious question. You've seen that snapshot of my career. So I'm a deep water sedimentologist and a quantitative modeler of the subsurface. Why am I talking to you about amber? So you give it a click, it'll take us through some reasons. So yeah, it all started, it was a hobby basically, started in Oman about 2008. When I noticed that you could buy amber online, and I bought some specimens, and I started learning the technique to take photographs of these tiny things within the amber. And it's something called focus stacking. And I shared the images I made online. And then I gave links to two guys. So one of them is George Poynar, Oregon State University. And he is famous because he had the idea that if a fly bit a dinosaur in the Cretaceous and was then encapsulated in amber, then the DNA of the dinosaur might be extracted from the fly, which is probably completely impossible, but it was a really bold idea. And of course, a start of a very nice series of films. So one of those images you see at the top right there, he published it in 2011, and we'll be looking at this in a moment with the new techniques, with the microtomography, and learning some more about it. So if you click again, the other person I sent the links to was David Grimaldi at the American Museum of Natural History, and he also borrowed specimens. And in 2011 as well, he published the thing on the right. So it's named after me, Burmacirtus reesmithi, a 100-million-year-old fly with strange wraparound eyes. So these are the two most famous people in the area of research into biological inclusions in amber. And click once again. And 2018, I published my first paper on amber with somebody called Andrew Ross at the National Museum in Edinburgh. And that's what I'll be showing you the central story from in the next few slides. And there's maybe one or two, yeah. So last year, publication with Juan Daza, Sam Houston State, on the earliest skink, a kind of lizard. So that is out in Nature Scientific Communications. And one final click. This is also last year. A nice team of entomologists and paleoentomologists did me the honor of naming that grasshopper, 50-million-year-old grasshopper on the right. So it's called Reesmithia Gorochovai in the tribe Reesmithini. So quite a productive hobby to have. And if you click onto the next slide, what's the purpose of the talk? So I'm going to show you how microtomography helps us acquire new insights, both into paleo environments and the interactions of organisms. And the first story is going to be about the Myanmar Cretaceous amber forest, which is pretty fascinating and overturned previous ideas. And then the second will be about the Dominican ant and a mealybug. So this is a symbiosis story. We'll be looking up close at that relationship. And the next one, please. And the final one will be about another Dominican amber beetle and its nematode passengers. So we've got one paleo environment story and we've got two symbiosis stories. And I'll show you aspects of the technique as we go along. And that is the three stories that we're going to look at visually. So the top one, number one there, amber grounds, that's a term I made up, by the way, borings and the amber forest. The one on the right is showing an ant carrying a mealybug. And then the one on the lower left is showing a beetle with its hitchhiking nematodes. So those are the three stories that we're going to be addressing. Next slide, please. Amber, so what is it? And I was reading this Roman, Pliny, writing about amber in 77, 77 of the current era. And this is what he wrote. Amber is produced from a marrow discharged by trees belonging to the pine genus, like gum from the cherry and resin from the ordinary pine. Liquid at first, gradually hardened, and it's a great summary. I really like it. So that's why I've put this very, very ancient view about what amber was. So you find it as far back as the Carboniferous, and you find it in the Triassic, and you find it with abundance peaks in the Cretaceous, the Eocene, and the Miocene. I think one more click will show you it in the present. I know different tree groups are conifers, araucarians, dawn redwood, umbrella pines, and also in the more recent amber from the Dominican Republic, from angiosperms, this plant called Hymenaea protera. I think there's a picture on the next click of the slide. And this is from the base of Kilimanjaro in Tanzania. And what basically happens there is when the tree is damaged, the resin is a repair mechanism. And when it gets buried, it polymerizes and transforms from that resin into amber. All right, so how do we go about documenting these inclusions in amber? And this is a snapshot from my Lightroom screen and photography. So when you take pictures at very high magnifications, the depth of field is very small. And the solution is what you're seeing here. And I started doing this in Oman, but with my camera held up against the microscope, and then just moving the focus of the microscope to everything I wanted to have in focus, and then using Combine ZP, a focus stacking piece of software. But now I can't show you, but it's just to my right here is my ultra macro rail. And that is a little box which does it for you. So you start at the top, and I typically have 50 to 100 images. And then when they're all stacked up, so when all of the images that have in focus parts are stacked up, you end up with these superb images like the warrior termites that you see there on the right, even with really minute details on the legs. So this is one is in the review process at the moment as a new species. Now we can go to the next slide. So that was a 2D approach. This is the microtomography. So this is our machine in Kuala Lumpur, and that is Dr. Hijaz Kamal. And you can see it looks like a box from the outside. It's a Zeiss machine, and it's got a radiation warning in Malay on the outside there. And that right-hand picture is showing us looking through the window. So on the left is an X-ray source, and we direct a beam of X-rays to our specimen, which sits on the stage in the middle there. And then there's a sensor there on the right. So what you do is you rotate the specimen through a full 360 degrees and take images at very small increments. And then you use software to combine all those different views from every different angle. And it provides you with a stack of slices of images. And the resolution is, it's quite often 5 microns, 10 microns, really tiny resolution, stack of slices. And then you can use software to interpret, and we'll talk about that later. So next slide, please. And this is just an example now, and we're going to be looking at amber examples, but this is just to show you the kind of interpretation that can be done. So if you click on it, it should move. And this is Malaysia's only fossil of a stegodon, and it's almost entirely still in the rock. So what I've done there is I've interpreted the enamel of that tooth. It's beautifully complete. And what I've done there is made the matrix, made the rock transparent or nearly transparent. So it was pretty fortunate. Look at the scale down there. It's a baby. It's a milk tooth, but it completely transformed the perception of the shape and the components of this tooth. So a student had interpreted it basically entirely incorrectly, but then now I could print out in 3D models at the proper scale, and that is now accepted for publication. And one more example on the next slide, and again, if you click it, it will run a little movie. So this is the ideal case. So what happens when you direct that, click on it to make it move, yeah. When you direct the X-ray beam, is the X-rays are attenuated when they come across something in the rock which is denser than the matrix. And this is the ideal case where we've got a trilobite, which is recorded in pyrite in a mud rock matrix. So if you click, yeah, here we go, now it'll go. So we're rotating the specimen, and we're going to start playing with transparency now. And yeah, so this is the perfect case. This is rather easy, and you can just use thresholding to isolate the high density, high attenuation object, just like that. It's not as easy with things in amber because you don't have that density contrast. But this is the kind of circumstance where it becomes very rapid indeed to make that kind of interpretation. So let's go to the next slide. Story number 1, the amber forest, the coastal, well, I put coastal, which is the punchline actually, because that wasn't known before. And let's go to the next slide. Myanmar, beautiful country, troubled country. I've only been there. I've been there twice actually. This was in a brief window before COVID shut everything down. But a really beautiful place to go. Let's go to the next slide. And so on the left, I've got Malaysia in the middle, Borneo in the middle. Up top left, east of India, you can see Myanmar. And also in the zoom over here, so you can Shwedagon in the south, Bagan in the middle. And you can see where I've labeled at the top, the Hukong Valley. That's where the amber comes from. And it is Cretaceous in age, about 100 million years old. So if we go to the next slide, we'll see what it was like 100 million years ago. So the red triangle there shows where the area where the amber was produced was. It's on the northern margin of the Tethys Sea, and a little bit north of the equator. And that's where that was. So let's go on to the next slide. And the Burmese amber is just incredible as a sample of the things that lived in a 100-million-year-old rainforest. And you can see a graph here of species described against time. So starting back in about 1999 and coming up to the present. In red are the six-legged things, the hexapods, and in blue is all taxa, total taxa. And you can see that we've got to 2,805 species described, and all of them beautifully preserved in amber. So it's really, really remarkable. And Andrew Ross, my co-author on the paper here, he keeps tabs on that, and every now and again releases a new summary of where we've got to, what's the state of play and what's being described. Let's go on to the next slide. And I'm going to just show you a few snippets now of a modern creature in a rainforest. This is Northwest Borneo, one of my favorite places to go over the past decade when I've been living in Malaysia. And on the right, so the yellowy orange ones, those are 100-million-year-old pieces of amber. So we have a whip scorpion on the left, Protheliphonus, and then immediately to the right is one from Myanmar, spectacular, complete, beautifully preserved. Yeah, on the right we have a centipede, large forest centipede. On the bottom, an example in amber. And we can click through these fairly quickly. It's just to show you what a remarkable capture it is. They look like they were alive this morning, but they're in fact 100 million years old. Tarantula on the left, spider on the right with multiple eyes looking at us. So if we go forward again. And I think if you click one more time, we get a close-up of the eyes. So you can see those eyes with a little touch of pyrite in the middle of each. And click again, we'll go on to a couple more. And there's a harvestman arachnid on the left, so modern and Cretaceous. And there's a damselfly there, modern at top and Cretaceous underneath. And a couple more. So next slide. There's a snail, terrestrial snail in the amber there with its operculum. So we go to the next slide. And a lantern bug, modern and very ancient. So beautiful sample. And this is a kind of lizard, a skink. So one from Borneo on the left and one from the Cretaceous of Myanmar on the right. That's something that should be published this year. It's an amazing, complete lizard and a piece of amber. Let's go to the next slide. All right, so the Burmese amber is often filled with this feature that you can see here. So you see these strange club-shaped objects. People often describe them as clavate objects. And yeah, what are they? So if we go to the next slide, we'll see people's previous ideas. So Grimaldi et al. 2002 is the best overview of this amber occurrence and all of its inclusions. And so David Grimaldi from New York offered the explanation there on the right. So if you look at the top, you can see that there's a cavity in the wood, and there are all these features lining that cavity, and he interpreted them as fruiting bodies of fungi, sporocarps, something like a mushroom. And in the middle, the resin has flown into that cavity and then has preserved all of those club-shaped objects. And then the bottom image shows it peeled away from the tree, and now it's a piece of amber. And he's used the right language. He says possible sequence for the preservation of sporangia. So it's not correct, but it was an idea proposed. If we go to the next slide, that was 2002. 2003, there's another paper, Poinar and Brown, and all the caution has gone now. So it's got a genus and a species, Paleoclavaria burmitis, and it's got a family name that's been stated, and it's described as the first fossil record of this group of fungi. And then on the right, there are some very specific details there, cystidia that are labeled, features that you find on the surfaces of mushrooms, basically. And I just took these views that these were fruiting bodies of fungi, just took them on trust for about 5 years. And if we go to the next slide, so the next slide is when I, out of curiosity, make a 3D scan. It's a scan of the very same specimen that I showed you a few slides back. And if you look on the left, you can see these club-shaped objects, and you can see that they're cutting across what in the middle there is a plant. So I wrote to David Grimaldi and I said, well, I'm seeing these cross-cutting relationships. Doesn't seem very consistent with fungal sporocarps. And then he nicely said, well, you have discovered my worst ever scientific mistake. And I suggested we might publish it together, but he didn't want to. And I've done that in the past when there's a revision needed. Just go back to the first author and say, shall we write this together? But in this case, it fell to me, and I teamed up with Andrew Ross to bring in some specimens from Edinburgh. So on the right are some images, beautiful 3D images, and it's got, again, a lovely density contrast, like the pyritized trilobite. So you can actually see the shape of those structures in 3D, shaped like little clubs. Yeah. So next slide. So we've had one observation, there's cross-cutting relationships. Here's another observation, not just cross-cutting, but they've got sediment fills. So you can see I've labeled there, coarse lower sand, very fine sand. Some of them were empty and have got calcite, sparry calcite fills, but they're clearly nothing to do with fungal sporocarps. Let's go to the next slide. And another observation, a third observation, some of those structures have got bivalves preserved inside of them. And these are very distinctive boring bivalves that land on a rock, or in this case, a hardened piece of resin. And then they bore in and live inside what's called a crypt. And in that picture on the right, if you look at the lower left side, you can see there's lots of whites there. And you can see there's lots of very fine structures. Those are the ratcheting marks that bivalve makes when it's excavating its crypt, its boring. So 3 observations, that's quite good. And they're no longer fungal sporocarps, are they? Next slide. 4th observation, sometimes the bivalves there do not cross-cut what's there before, like the little leaf there. And they leave behind, if you look, maybe jump to the lower left image there, you can see there's an irregularly shaped structure. Sorry, not lower left, lower right. There, yes, you can see the bivalve at the end. So what's happened here, it looks like the bivalve has pushed into resin that's still soft, which is really interesting. And it was really interesting to my co-author, Andrew Ross, because he thought the borings meant that the amber and the insects inside could be much, much older. But what this is saying is it looks like it's been trying to bore into something that's still soft. And if we go to the next slide, we get support for this idea. So this is over in Florida, and it's a swamp forest. And what you can see there is that there is resin from one of those trees, a swamp cypress, that is traveling down the tree and into the water. And in the water, it hardens very, very slowly. So it makes a skin, but that skin can still be penetrated by aquatic organisms. So things get inside the soft. It's something which is kind of hard on the outside, but yeah, you can see all the little white arrows there. That's where creatures have gone inside and are destined to become inclusions in amber once that's been buried and aged and had time to polymerize. So it seems to work. And because those bivalves, they die in fresh water, so they can live in normal marine salinities, but you most often find them in brackish, so low salinity, brackish marine settings. This tells us now, doesn't it, about the paleo environment. We're looking at a forest which was in close proximity to seawater, at least brackish seawater. So from these little observations on tiny pieces of amber, we can make an inference on a much larger scale. So let's go to the next slide. And just after this paper was published in 2018, several other papers piled in with more evidence of aquatic organisms, and specifically marine aquatic organisms in the amber. And this one was the most spectacular by far. So it's an actual ammonite in amber. So you must be in a coastal setting for this to happen. It has no soft parts. It's obviously been a little bit abraded, but an incredible amber enclosed ammonite, which also very nicely confirmed the age of around 100 million years. So yeah, we have totally overturned the ideas of fungal sporocarps and all those inventions of new names and family names and specific details. And we've got some information now about the larger context for that forest. So it's a very, very well cited paper, because that provides the context now for all of that rainforest biota that we're looking at in the amber pieces. One more slide. And so this is somewhere that I would come every couple of months. This is, we're looking at Borneo now from the north. So that's Brunei. And you can see the Barham Delta out, that spiky protrusion out to the right. There's this incredibly sinuous, yeah, the Barham River that comes out there. And if you look to the kind of center towards the left, that's Brunei Bay. You see that large bay there, yeah, and to the left. So if we click once more, I think we'll get a close-up of that area. So that's the kind of thing that I envisage, where we have the forests very, very close to, you know, coming right up to the coastline, basically. And that's good fun, because I was thinking, Oman, amber in Oman, I never heard of any, I never saw any, but I did think that the Rusail might be a good candidate. And I've looked in the Rusail in the Coley intervals without finding it. But there are mangrove palynomorphs in the Rusail. So again, it's evidence of a coastal forest, but maybe one that in that environment just didn't produce the resin like it did so abundantly in Myanmar. All right, so let's move to a different part of the world now. So we're going to go into the topic of symbiosis stories. And this is just a view which shows top left a bee laden with pollen on its legs. So there's one mutually beneficial symbiosis between the plants and the bee. Middle top is the one that we'll be talking about, the ant and the bug. Top right's quite funny, because it's a kind of fly. But when you look up close in that lower right image, you can see it's got this pseudoscorpion just hanging onto its leg. And that's really common, that there are little creatures that catch rides with bigger creatures to get translated to somewhere that's new and more favorable. And the lower left image is another example of that, with the little tiny nematode worms hiding under the wing covers, the elytra of that little beetle. So let's go over to that part of the world. So the ones we're going to be looking at are much younger than the Cretaceous things that we've looked at so far. So these are going to be about 15 to 20 million years old from the Dominican amber. So if you click again, it'll probably say, yep, that's the title. Click again, and it should take us west. Click again. And we go for a little journey. Whoa, a man along the way over the Atlantic. And here we are now in the Dominican Republic. And La Cumbre is one of those places where the amber is found. All right, next slide, please. Okay, so the first example, something called trophobiosis. And there's a definition down there, symbiotic association where food is obtained or provided. And the one that provides the food is called the trophobiont. And you probably all know this about ants and aphids, or ants and mealybugs, but they essentially farm them and feed off the honeydew that they produce. So let's go next slide. And this is some examples from the modern. And you can see that it's a female ant. So she's a queen, and she's taking a little mealybug to a new place to start a new colony. And she's just kind of holding it more or less gently in her mandibles there. So that's in the present. And if we go to the next slide. Just to show, I'm not a biologist at all, but if you look down that left column, those are all different species of the same kind of ant, Acropyga, that we just looked at. And then the second column are all the different bugs that they farm. So this is a bit of a revelation to me that there are so many symbiotic associations between, even within the one genus of ants, the one kind of ant. So let's go to the next slide. And this was first noted in amber, 2001. Very, very rare indeed. But that's a little bug there that you can see, the thing called Electromyrmecoccus inclusus. And a couple of specimens actually had the ants carrying the mealybug in the mouth. So let's go to the next one. Next slide. So I said they were rare. So how rare? Click again. And once again, we have David Grimaldi mentioned here. And he looked through 30,000 pieces of amber and found 3 examples. So they are that rare. And this is one of the things that I bought online back then when I was living in Oman without knowing what it was. So here are some photographs, left and right, and made with that focus stacking technique that I talked about before. So you can see all those incredible little details, like the little hairs on the end of the mealybug. And that middle image is a 3D extraction using software called Drishti, which means vision in Sanskrit. And that's made by AJ Limai of the Australian National University. And that is a lot of work, because it's not like the pyrite trilobites where you just say, just show me the high attenuation, high density stuff. Here you have to go and do quite a lot by hand. But the benefit is that now you've got a 3D interpretation of the thing. You can go in and measure it very precisely, and you can look at it from any angle that you want, which you can't from the piece of amber. So let's click one more time. And this is the result of me going in and measuring it and comparing it with an earlier paper, which in green shows the size ranges. So you can see they're all very small measurements. They're all in millimeters. But there's the total length, the length of the head, the width of the head, the scape, which is a long segment in the antennae, the mesosoma, the gaster, like the abdomen on the end. But I have in the big blue circles there got the most precise measurements, because I can just click at one end of the thing I want, click at the other end, and I've got the measurement. Whereas the other ones are all taken from photographs, and incredibly often with 3 decimal places, and you think, well, hey, you don't even know that you're looking in the right angle. But that's a benefit of these 3D images, of course. Next slide, please. And if you click again, so this one I made very recently with a different piece of software to see if I could do it quicker and better. This is something called 3D Slicer. So if you click, it should rotate. And parts of this were easy. So you can see the bubbles are there in blue. And the interpretation is the whole problem is a bit like 3D seismic interpretation, where there are volume interpretation techniques, and then there are manual techniques where you have to override and fill in. But there I could just paint the bubbles, which are the lowest density, just say that's a bubble, and then paint the amber, and that very nicely picks out the bubbles. But the ant itself and the mealybug, they were a lot more difficult, because they're a mixture of holes and pieces of tissue. And also, despite very high resolution here, I still don't have the resolution of the photograph where I could see all the little hairs on the tail end of the mealybug. Okay, so that was one symbiosis story. And now here, yeah, it's good. Next slide is good. And here's the other one. And again, it's a tiny piece of amber. It's got 2 beetles in it. And George, go ahead. Yep, that's fine. So George Poynard published it. That's his photograph, and it's pretty good. But we could basically look at this from underneath or from above, basically. And this is another specimen that, out of curiosity, I then put into made a 3D scan of. And if we go to the next slide. spent a lot of time again. So that's a photograph. And you can see also that the amber is not all transparent, is it? It's quite murky in places. So we've only got 2 viewpoints, and there's quite a lot of obscuring discoloration. So if you click again, you can see the little nematodes there. So this is the result of me interpreting quite often on a laptop in tea rooms in Kuala Lumpur with people peering over my shoulder. What's that? But in pink there are all the nematodes. And you can see the scale, it's only 1 millimeter. And it's quite a dramatic story. So they have all climbed aboard. They're mostly juveniles. There are a couple of adults there, and they've hidden beneath the wing covers. And the beetle has gone off flying somewhere new, which is good for the nematodes. That's what they wanted. But then hit some resin, some tree resin, and was stuck. And a globlet of tree resin has engulfed both the beetle and all of the nematodes. And then the little nematodes have headed off swimming. And so it looks really dramatic, but then on the scale is less than 1 millimeter. So you can see that they swam as far as they could, but none of them made it out. So that's the story there. But remarkable, isn't it? The 3D image. You can see the eyes on the beetle. And if you look at the next slide, one of the beauties of this approach is that we can control the transparency. So we're looking straight down. You can see the beetle, eyes, antennae, legs, and you can see there are these 2 bubbles of air which became trapped under the wing covers when the insect was stuck in the resin. And now we can count many more nematodes than we could possibly see optically beforehand. All those little pink things, and there's a very distinctive little sigmoidal one that went down the bottom, which I think was the one that got the furthest, 0.6 of a millimeter. So sad, dramatic, but rather remarkable, the images that we can obtain. And I think we're probably coming quite close to the end of the talk now. So let's go to the next slide. Yeah, so that's a visual summary. So we've talked about techniques, and we told a story about observations on small pieces of amber, leading us to understand that forest was a coastal forest, and that's where the trees producing the resin that contains those thousands of different species of beautifully preserved rainforest creatures, and that's where it was. And I showed you then a possible analog in the form of Northwest Borneo. And then we talked about the Acropyga ant queen carrying her mealybug in just a remarkable image. Not quite as old as the things we talked about with the Myanmar one, so maybe 20 million years old. And then the last one we talked about was the hitchhiking nematodes. So lots of fun, and the kind of science that's done out of curiosity and play. Like if we look at this with new techniques, what can we see that's new? Or what can we measure that's new, and what can we infer that's new? So if we go to the next slide, it might be conclusions. Yeah, so let's see. So first of all, it is I think you'll agree, microcomputed tomography, a very powerful tool for visualization and analysis. And I've shown you 3 amber cases, but I use it for lots of other topics, including crystals, so crystallography, other pyritized fossils, including an incredible vampire squid from the Jurassic, which I'm just finishing up at the moment. And then in the story, so we saw cross-cutting relationships, we saw sediment fill, we saw bivalves, and that completely overturned the previous interpretations of fungal sporocarps. And those are just basic geological observations, enhanced and facilitated by having a complete 3-dimensional image of those specimens. And the evidence for the boring bivalve spat colonizing the pebbles before they hardened strongly suggests that proximity of the resin producing trees to the brackish marine water. And the Florida example is a beautiful support for that. And I've mentioned with the ant mealybug story that we can make very accurate measurements indeed. And then finally, we could just choose whatever orientation that we want. Often not an option for optical observations, and particularly not an option for specimens like that primitive elephant tooth that's still in the rock, that hasn't been exposed from the rock, so completely invisible to optical investigation. And I think that's all that I have brought for you this evening. And yeah, thank you very much for being patient at the beginning as we wrestled with both Zoom and Teams. And I'll be really delighted to hear your thoughts and questions.
Thank you, Ro, for the interesting presentation of the Amber history and very interesting photos to present about many types.
And we
Now we'll
caught up on
move.
time, hey?
Yeah, we still have more time for the question.
Yeah.
Please, if you have any question, just raise your hand. I will open the mic for you to ask.
So it's quite funny because you've seen I've had a third of a century subsurface career with lots of 3D modeling, lots of seismic interpretation, and it's similar, but at a massively tinier scale. So now we're on a microns to millimeter scale, but the problems are quite similar. Extract the information from the 3D data set using the best techniques you can, and then interpret it to understand what was going on.
Any question is coming? Bro, you mentioned about the Amber could be in Oman, in Rusail, I think.
Yes.
Any plan for you or any from your team to do research or something in Oman during the next few years?
There is no plan, but I would love to. Yeah, because I'm going to have to come back anyway as a tourist. I've stayed away too long. And those outcrops are quite convenient, aren't they? Close to the university. But I was also going to ask any of you, do any of you know of times when people have found amber in Oman?
I never heard about it in Oman.
Yeah. Because Eocene's a good time. So all of the Baltic amber is Eocene. So here in Yorkshire,
Yes.
where I am right now, on the coast, we have beautiful Jurassic exposures, but there's almost no amber production in the Jurassic. So I'm still going to look when I go out to the coast. But Eocene should be better. But there's always a chance that if it was mangrove forests that we're talking about there, that they were just not the right kind of tree, the kind of tree that bleeds resin profusely, like we have evidence for in Myanmar.
Why it should be more would be available in Eocene?
Now that
Exactly.
is a really good question. And I've seen that there is a lady called Layla Saifullah who's been pursuing that question. Why do you find quite a lot of amber in the Triassic, none in the Jurassic, lots and lots of amber in the Cretaceous, almost none in the Paleocene, lots in the Eocene, almost none in the Oligocene, and then lots in the Miocene over there in the Dominican Republic? And the answer isn't very obvious, but the way to go about it is to think, well, what's the reason for trees producing lots of resin? And it means in essence that they're damaged. They're being damaged a lot. So what are they being damaged by? It could be insect damage. So it could be things feeding on the trunks, a kind of competition between being a food source and then finding a defense. And then another kind of damage can come from forest fires and burning, basically. And that's the kind of thing that would correlate with climate change over those, well, yeah, back to the Triassic, so over 200 million years. So I don't know what the answer is, but it might be a combination of those two factors. So, there's the proximate cause, which is damage to trees, a lot of damage to trees, and trees with an ability to produce lots of resin to heal themselves. And yeah, times when there was a lot of trauma being done to the trees, either through living things or through fire. Really interesting question. I'm going to read her work from now on.
Question is coming from Marina Gomez. She said, thanks, Alotro. Super interesting talk. Have you heard of any similar of the early Cretaceous amber from Cantabria in North Spain?
In North Spain?
Cantabria in North Spain, yeah.
Yes, though I don't know it well, but I do know people that have been working on that. So Andre Nel, he is someone that I've published with in the past on much older things on Carboniferous big insects, and he had some papers, I think, on that. There's also some Cretaceous amber. I think it's also Lower Cretaceous in the Isle of Wight, which is possibly the only amber that I'm aware of in the United Kingdom that's in situ, that's not been brought by currents and waves and washed up on the coastline. But I don't know anything specifically about that occurrence. It might be, this is only a vague memory now, but certainly the French Lower Cretaceous amber was nearly opaque. So they used this same technique of an X-ray microscope to image the inclusions because they couldn't see them optically. So that's when this technique becomes very, very powerful indeed. You need it. Otherwise, you don't know what's in there. And yes. The Spanish occurrences, I don't know. I'll have a browse after this. I'll have a quick search and see what I can find. Yeah, it's quite...
Thank you.
Yeah.
For answering the question. Is another question I think is coming from me. Oh, okay. She just said can be used in bitumen. Thank you.
In bitumen.
Can it be used in bitumen?
Why ever not? I've never heard of that being tried, but what you would need to be successful is to have a density contrast. So whatever was enclosed, I'm kind of thinking about Alan's, he has some little, what were they? They were little arthropods, I think in bitumen.
Bitumen.
In Oman. And I'm forgetting the details now. But yeah, if there's no density contrast, you won't see anything, basically. So, and you might be in the case where I was here with very low density contrast in the amber, where you end up doing a lot of manual work, so a lot of manual interpretation line by line, like in seismic. Yeah. Yeah. Is there anything behind that question?
There's no more question. Did you mind if you shared your email, or maybe I could share it in the chat box if someone would like to ask?
Yes, that is fine. Yeah.
Okay, I will share your email. If someone has any question, please.
Yes.
Communicate directly to Ross Smith. I think another question. Oh, okay. Okay,
Yeah.
no more question.
Yeah.
No more question.
So in Oman, do you know of people using